In this lecture, we're going to look at how the chemical agents that were discussed in the last one are responsible for allowing sterilization or disinfection, depending on what they do. And then we'll look at some of the specific chemicals that are used in sterilization and disinfection. So the first way that we're going to talk about that these chemical agents work is through protein disruption. So many antimicrobial materials work because they cause chemical reactions that damage the components of a microbial cell. Primarily, or often, frequently, they are proteins. We also will see how lipids can be disturbed or damaged as well. But when we disrupt proteins, that process is referred to as denaturation. And we haven't spent a lot of time talking about this, but if you think of what a protein is, it's a chain of amino acids. So if these little circles that I'm putting into a little pop bead necklace style structure here are amino acids, then that would be a very, very, very tiny, in fact, we wouldn't be able to call that molecule a protein, it's so small. But these long chains that have thousands and thousands of amino acids in them, they form hollow tubes. So the chain becomes like a spring, a hollow tube. And then that hollow tube, and that's kind of what they're showing here, that hollow tube folds up into a really complex three-dimensional structure. And it is that complex three-dimensional structure that gives the protein its function. And so lots and lots of chemicals will act because they cause the protein to unfold into a way that it loses its structure. So if you think about an egg white is almost pure protein. It's, well, there's what I should say, I should take that back. It is protein dissolved in water. So it's mostly proteins dissolved in water. And if you heat that protein, then heat can cause that unfolding. And we can permanently denature a protein with heat, for instance. And that's what happens when we have a fried egg. The egg white goes from being loose and thin and runny to being a solid that is clear, that is no longer clear, but is instead it's an opaque white. So some chemical reactions may only temporarily denature the protein, but it can temporarily denature the protein long enough to kill the cell. And then the protein will renature or reform into its specific structure. That's why warming or heating milk is important. So like if you were going to make yogurt at home, then you would heat up the milk, you would almost boil it. That heating of the milk would temporarily denature the proteins that are in the microbes. And then when the milk cools down again, then it would restore its shape and restore its function and restore its flavor too. And if you remember in class last week, we were talking about box milk, not dry milk, but the milk that is in the cartons that sits on the shelf. That milk has been so heated that it permanently denatures the proteins. Okay, so the reason that protein disruption works is because we find proteins in the cell membranes and we find proteins inside of the cell that work and function as enzymes. And so if we can disrupt those proteins long enough, then that can kill the cell. Either weaken the cell membrane so that the cell dies, or denature the enzymes long enough that either they are permanently denatured and can no longer function, can't allow all of the chemical reactions that we think of as metabolism in the cell to occur. And so if the change to the protein is permanent, then we describe that function of the chemical as being bactericidal. If it is a temporary change, then that produces a bacteriostatic effect where it kills back. We can think of this as like the difference between sterilized and sanitized almost. So it's the same kind of idea. bactericidal effects, kill the proteins. Kill the bacteria because they destroy the proteins. Bacteriostatic effects really slow down the ability of the bacteria to grow. So, what are the ways that this kind of denaturation of proteins can occur? We can do it with high heat, with extreme pHs, so either at the acidic end of the pH scale or the basic end of this pH scale. We can do it through reactions that cause hydrolysis. That's where we break the protein into smaller chains. So this actually goes and breaks. So again, if this is our chain of proteins, hydrolysis would cut the protein into smaller chunks, and then it would no longer be able to do its protein function. Oxidization, which is where we either add oxygen to the structure of the protein or remove hydrogens from the structure of the protein. So oxidization occurs with hydrogen peroxide. That's why if you put hydrogen peroxide on a cut with blood or tissues, you see all that bubbling. And with halogens like chlorine, like you find in bleach, or iodine, like you see in a material like betadine. You can also denature proteins by forcing them to interact with heavy metals, stuff like mercury or silver. These are example heavy metals. Or through a process called alkylation. Alkylating means adding a methyl group. A methyl group is a carbon with 3 hydrogens. So here's the carbon. It has space for hydrogen to attach to it with a chemical bond. And then this chemical bond is available to attach somehow to the protein. You attach methyl groups like this or other similar groups to the amino acids in a protein, and you alkylate them, and you can denature the protein, change its shape. And again, as you change the shape of the protein, you change the protein's function. It can no longer do the function that it was doing in the previous shape. And so dyes like the stains that we use in staining cells, formaldehyde is another alkylating agent. There's a bunch of different alkylating agents that can be used. So let's think first about disruption of the cell membrane as a way of destroying cells, of either killing like bactericide or bacteriostatic effects. We can disrupt the membrane by denaturing the proteins that are found in the membrane. And we can also disrupt a membrane by dissolving the lipids that are in the membrane using a surfactant. The way that surfactants work, if you have a glob of lipid, let's say this is some phospholipid like you would find in a cell membrane, surfactants work because a surfactant has one end of the molecule that dissolves in lipid, so that's the lipid dissolving end, and the other end of the molecule that dissolves in, dadgummit. Okay, there we are. The other end of the molecule that can dissolve in water. So this is water dissolving. And so we can surround our lipid with these surfactant molecules. The lipid dissolving end interacts with the lipids, and the water dissolving end will interact with the water molecules that are all around in the fluids. And in this way, we can disrupt the, if you recall, the membrane is built of phospholipids, and they have their hydrophobic tails pointed toward each other. So now we can interrupt this hydrophobicity by sending all these surfactant molecules in there, and that can poke holes in the membrane. So hopefully that makes sense to you. If not, then send me an email, and I can try to come up with a small video or something like that that talks specifically about how surfactants will surround individual lipid molecules or even clusters of oils. And so we use detergents like laundry detergent or dishwashing detergent. They work to to get oils off of our dishes or out of our clothes because they act as a surfactant. They surround these oily molecules, allow them to dissolve in water where normally oils don't interact with water. They separate in water. And then we can wash them out of our clothes and away down the drain. Alcohols also act as surfactants, and quats, what are called quaternary ammonium compounds, such as benzalkonium chloride, that's found in Lysol, in antiseptic soaps and antiseptic towelettes. They act as surfactants. Merthiolates act as surfactants, and other quaternary ammonium compounds are all surfactants. Okay, so two ways of disrupting membranes. We can do protein denaturation, and we can use surfactants to break up the membrane. Another way that we can help to destroy or produce bactericidal or bacteriostatic effects is to disrupt other cell components that are found in the cell. So alkylation, as we've said, can disrupt the function of the nucleic acids. It can disrupt the function of the energy production systems. So the photosystems that do photosynthesis and all of the processes that make ATP from glucose can be disrupted by alkylation. Some of the stains, for instance, crystal violet, that we have used to stain and dye cells, will interfere with the production of the cell wall of the bacterium, and so that messes up with the ability of the cell to grow and reproduce. And sometimes the end products of fermentation, like lactic acid and propionic acid, they will inhibit fermentation from happening. So this is kind of a feedback inhibition. So I should have had a close parens right there. So add a close parens in your notes. So these end products of fermentation are alcohols or acids, and they will feed back and kill off the bacteria that are causing fermentation to happen. And we describe that as negative feedback or a feedback inhibition cycle. Some chemical agents also work to inactivate viruses. So alkylation will disrupt DNA and RNA that are found in bacterial cells and more importantly here, in viral cells. And while protein denaturation doesn't always destroy a virus because that protein shell that is, the capsule that's around the virus particle, it's pretty resistant to denaturation many of times, but using detergents and alcohols around membrane enclosed, so the enveloped viruses, can render those viruses inactive. So we can't always rely on protein denaturation to inactivate a virus, but if it's an enveloped virus, then those molecules that can surround lipids will have their effect. Okay, so what are some of the specific chemicals that we use as antimicrobial agents? Well, soaps and detergents are one of them, and they are really good at removing oily substances and dirt because of their surfactant capacities, especially when they're accompanied by vigorous scrubbing. And as Lister showed early on, this is the cheapest and easiest means of preventing the spread of disease. So this is where Lister was busy trying to get all those doctors to wash their hands between doing autopsies and going and helping the women to deliver. And then if you follow that soap and detergent with a 70% alcohol rinse, then you have an especially effective means of bacteriostatic effect of hands, on the hands. Soaps also are alkaline, so they have a basic pH, and frequently that will interfere with the proteins and can even kill Streptococcus, Micrococcus, and Neisseria genuses or genera of bacteria. And the detergents that we use for both laundry, for dishwashing, and for other cleansing, they can be either cationic or anionic. So these detergent molecules have either a positive charge or a negative charge. And it turns out that the ones with a positive charge, the cationic detergents, are most effective against bacteria. And we presume that that's because the membranes of bacteria and other organisms have a negative charge. So cationic, positively charged surfactants are the most capable of disrupting membranes. Okay, so that's the soaps and the detergents. Acids and alkalis will also do this. As we've mentioned, soap is mildly alkaline, which helps to aid in microbe destruction. And then organic acids that lower the pH sufficiently can also inhibit the activity of, as we've said, fermenting bacteria. And so we often add lactic and propionic acids to breads and other baked goods as they're being made, because this has a bacteriostatic effect and acts as a preservative. Benzoic acid, again an acid material, is used to prevent fungus from growing in sodas, in margarines, in ketchup, and other moist and wet foods. And sorbic acid and other sorbates, some of the salts of sorbic acid, are used in cheeses and other moist foods to help as a preservative. So these organic acids, and they're organic because they have carbon and hydrogen in them. Most organic materials also have oxygen. So if they have carbon, hydrogen, and oxygen, we refer to them as organic materials. So lactic, propionic, benzoic, and sorbic acid all have their bacteriostatic effect in foods because they lower the pH, make it more acidic. Heavy metals, as we mentioned, can be used, and some of the heavy metals that are especially effective against bacterial growth are selenium, mercury, copper, and silver. Silver nitrate eye drops were, back in the day, back when I was born even, all children were administered silver nitrate eye drops who had gone through a vaginal birth as a preventative against gonorrhea infection and the risk of blindness that having gonorrhea infection of the eye could cause. Silver ions are now being incorporated into fabrics and into plastics because of their bacteriostatic effect, the antibacterial properties that they have. And the way that these silver ions work is that they interfere with cellular respiration in bacteria, especially if they're gram-negative. Remember, gram-negative have a thinner capsule. The silver ion can penetrate into the cell, find the mitochondria, and prevent the cellular respiration processes that make ATP from happening. Mercury, a heavy metal, and of real concern because of mercury contamination of waterways as we've been dumping pollutants into the water and into the ocean. But we've used, for a long time, we used mercurochrome, like old school mercurochrome, had mercury in it. And merthiolate, also called thimerosal, that they've been used as skin disinfectants. We're seeing, the new merthiolates are mercury-free, and thimerosal has been, they used to use it in vaccines and in eye contact lens solutions and eye drops. And so we're seeing less and less thimerosal being used as well, just because we recognize that mercury has some real issues. And if you can get mercury poisoning, which affects nervous system function. Phenylmercuric nitrate and mercuric naphthenate are also used in laboratories as lab surface disinfectants. And then selenium sulfide is used because it's antifungal especially, and we see selenium sulfide in shampoos because people who have heavy dandruff, it's frequently because of fungal infections of the skin of the scalp. And so it's found in these anti-dandruff shampoos. I'm not sure if Head & Shoulders uses selenium sulfide, but it would be like a Head & Shoulders type of shampoo that would have selenium sulfide in it. And then last of all, copper sulfate can be used to control algal growth. But copper sulfate has some environmental hazards as well, so it is no longer being used that way. Okay, some other chemicals that have antimicrobial effect are halogens. These include chlorine, iodine, and bromine. So we use hypochlorous acid. That's the chemical kind of bleachy smell that we associate with swimming pools and in drinking water to help control bacterial growth. It's also found in bleach. Bleach is sodium hypochlorite. It's the salt of hypochlorous acid. And you know how heavy an oxidizer bleach can be. You know, it'll oxidize the dyes in our clothing. It'll oxidize cells, and it kills fungi and bacteria on surfaces. But the problem, or one of the issues with using bleach, is if there's a heavy growth of organic material on a surface, it can completely inactivate all of the hypochlorite that is in the chemical as it kills, and it can leave some organic material behind, including bacterial growth. Chloramine is a combination of both chlorine and ammonia, and while it's not as effective as other chloride compounds, it doesn't have the same odor and taste issues that we associate with using bleach or other hypochlorous acids. Iodine is found in betadine and isoden, and it's used as a surgical scrub. If you go backpacking and don't have a filter that would remove any, say, Giardia or other bacterial like E. coli from the water that you collect to drink, then you can use iodine tablets. It's a form of iodine that if you let it set for about 20 minutes, it will have a bacteriostatic effect in the waters that you're drinking. Although it also leaves a kind of unpleasant iodine flavor to the water. But if you're really thirsty, hey, you'll be happy to have that iodine water. And methyl bromide, which has the bromine atom in it, can be used to fumigate soils, like in a nursery or some other place where they're growing plants. You use methyl bromide in a gaseous form to fumigate the soils and kill off the fungi especially, but also the bacteria that are in the soils that you're trying to grow the plants in. Alcohols and phenols, these are specific chemicals that are used to both denature proteins and or dissolve the lipids that are in the membranes. So ethanol and isopropanol can be used as a skin disinfectant. And interestingly, 70% alcohol is more bactericidal than 95% alcohol. So you need that extra water, that 30% water, to help allow the alcohol to penetrate and to kill or to act as a surfactant on the membranes, or to penetrate into the cell and act as a denaturing agent. So a 10 to 15 minute soak in 70% alcohol will sterilize hard surfaces like combs at a beauty salon, or thermometers if you're using an old school glass thermometer at a doctor's office. You give it a good 15-minute soak and you can sterilize a surface. Shorter soaks are bacteriostatic. Phenols and phenolics, these are special organic compounds that also disrupt membranes and denature proteins. And there's a number that we use in both the medical field and in other places. So amyl phenol, it's called Amphil, is used on skin, on disinfecting medical tools and hard surfaces. Orthophenylphenol, that's the active ingredient in Lysol, is another molecule that's similar to amyl phenol. Cresols, like what we find in creosote, have an antimicrobial action as well. And so we use creosote on like railroad ties and telephone poles that are made of wood. Chlorhexidine gluconate, that's the active ingredient in Hibiclens, is used as a surgical scrub, and it has effect despite lots of organic molecules being around. So where bleach is ineffective when there's a lot of organics in the material, chlorhexidine gluconate in Hibiclens is very good at allowing disinfection. And triclosan, this is another phenol that's added to plastics to produce these plastic surfaces that have an antimicrobial effect. And they are effective on bacteria, but not fungi. And an issue with triclosan is that it produces bacterial resistance. So having lots of triclosan in our environment is going to produce triclosan resistant bacteria. And so other methods would have to be used then. Some more specific chemicals, oxidizing and alkylating agents. So we talked about oxidizers like hydrogen peroxide, that's H2O2. I love that the name tells you exactly its components. If there's one hydrogen per every oxygen, then you've got a hydrogen peroxide. And it releases highly reactive superoxides of O2 that has lost, or that has gained an electron, I'm sorry. So O2 minus is this superoxide, and it will quickly destroy proteins, but it's also quickly inactivated by peroxidases, enzymes that are found in bacterial cells and body cells. Potassium permanganate is another oxidizing agent that's used to disinfect surgical tools and skin. And then there are alkylating agents. Remember, this is adding a methyl group, that's CH3, or some other similar molecule to a protein or to a nucleic acid. But when we disrupt nucleic acids, that can sometimes then lead to development of cancers. So alkylating agents shouldn't be used where they can contact human cells. So formaldehyde, we use formaldehyde as a preservative in biology because it will prevent bacteria and fungi from growing. So when you do dissections, frequently the material has been fixed with formaldehyde, and that will help to prevent bacterial growth. Glutaraldehyde is another one. If you expose materials to glutaraldehyde for 10 hours, then that will even kill off the endospores in cells. Beta-propionolactone, there we are, is used to inactivate viruses, and so it sterilizes the vaccines and prevents viral contamination of vaccines. And ethylene oxide is used as a gas, and you can pump ethylene oxide into a chamber and then use that to sterilize, because it penetrates materials. It'll sterilize rubber and plastic and fabrics. But before you can use it, you have to thoroughly air out these materials because ethylene oxide is very toxic and is explosive. So super reactive material. Okay, so finally we're to the end of this whole list. So dyes and other agents that can be antimicrobial. Acridine is a dye that's used to disinfect wounds because it interferes with bacterial DNA replication. Methylene blue can inhibit bacterial growth. That's another of the stains that we use. And crystal violet, also known as gentian violet, it, as we've said before, blocks cell wall synthesis. Plant oils like thymol, it's a derivative of thyme, the herb thyme. Eugenol is a derivative of cloves, the spice cloves. So there are many plant oils. These are just two examples that have a disinfectant or bacteriostatic effect. And then sulfites and nitrates are frequently used as food preservatives and in helping to preserve wine. So sulfites, sulfur dioxide, and sodium nitrate is used in preserving meats. So lots of canned meats or or like salamis and bolognas, those kind of sliced meats that you buy at the deli, may have been cured or preserved with sodium nitrite. And there are issues with nitrates and nitrites and sulfites as well. So people have reactions to them, and they can have, they're associated with some diseases. Okay, so there you have it. All the ways that chemicals can be used as disinfectants or bactericides, and some of the specific ones that we use in different arenas. You will know what to do if you have any questions. Send me an email or give me a phone call in the office, or come and see me during office hours. I love to see you and hang out with you.